Okay, let's start. Hi everybody, I'm Nicolas and I'm from Montpellier, France, and I will present you a lot of things about mandrills, about mate choice, about sexual selection, but the key point is about generative artificial intelligence in ecology. Well, my purpose for the end of this talk is you will able to understand what happened to this little guy. Okay, those mandrills are something special. They don't exist. They are purely artificial. To explain all this mess, I will begin to do a big presentation of general concept of generative artificial intelligence. To, in a second time, explain how I build, modify, and measure mandrill faces to do behavioral experiments with mandrills. Okay, first, very quickly, what is a neural network? It's a mathematical model. So you have data. For example, it can be tabular data or numbers or an image, because an image it's a matrix of numbers, so it's okay. You put it on this neural network. There is just an amount of adjustable parameters, and each one have to be trained to put out an output. And this can be a probability, for example, for a task of classification, but also be synthetic data. Okay, take well, artificial intelligence is a really big thing with a lot of parts. Machine learning is one of those things. When you do machine learning, you have to train computers to learn about data. And deep learning, previous slide, is when you use neural network to do machine learning. Okay, and the smaller part is deep learning models to generate data that we commonly called generative artificial intelligence. There is a lot of a model to do that. For example, autoencoders, I don't detail, diffusion models, I don't detail, and the one I use, generative adversarial network, also called GAN, made by two parts, the generator and the discriminator, and I will explain this in details. The purpose of the generator is to create fake realistic data. And the purpose of the discriminator is to say if an image is from a database of real faces or real images, but faces of mandrills in my case, and or if it's from synthetic images. Here, this is pure noise, so it's easy for the discriminator to say it's fake data. Here, it is better. You can see a pattern and colors of a real data, but for the discriminator, it's not enough, and it also say it's false. But after 100 and 100 steps of train, the generator begin to be good, and it is able to generate realistic synthetic images. And for the discriminator, it's too hard to say it's fake, and it say it's true. So the generator is ready to create realistic synthetic images. Okay, this is the principle of GAN. And it was created 10 years ago, but there is a lot of improvements those last years, and I use this particular one, Seigen tree. Back to mandrills. So you can see two mandrills. One, we don't really know if they are angry, if they are friends, but there is a long-term study, the Mandrills project in Gabon, the purpose of this project is to discover things about mandrills. So they do a lot of things on field. But the one I'm interested is there is a big database of faces of mandrill, thousands and thousands of faces. Better. Field assistants take pictures every day, and because of that, we have a training database to do our computational vision work. Okay, those ones are reals from the real database. Okay, if I do that, that's my work is to answer to a problem with correlative field study, because in field we have a lot of confounding factors hard to control, But if we do an experiment, it's better because we can control confounding factors. To do that, we can modify an images with computer vision. So here is just an example with Photoshop, but you can see this is the same individual, but with different colors. So we control confounding factors. There is just the color would change. And with that, we can control confounding factors. This was just a toy example to explain for you. A second one with numbers. I train for this example. I train again just with numbers to create a space like for a PCA but non-linear. And in this space, you can see there is a continuum of numbers. So there is 7, there is 3, there is 1, and we can find an axis of how an image, the image of a number is a 7. So on the top left corner, this is definitely a 7, and there is a continuum which it's became to be less and less a 7, and on the bottom corner, this is not at all a 7. So if we can do this with numbers, we also can do this with mandrills. So this is also a space. The red dots are female faces, the blue dots are male faces, and we can find with a support vector machine a vector who separates those two clusters. So that's what I do on the space from the GAN. And I take the orthogonal axis of the support vector to have an axis of addition of mandrill faces. I will illustrate this with an example. Okay, I take my axis, I project on it all the faces of my database to have a distribution of facial representation of my mandrills. Okay, I have a female distribution of a male distribution. With that, I have descriptive statistics like a mean or standard deviation for each sex. And this is the key point of my presentation. If I take a face of mandrill, if I project it on the axis, I can edit it to make it less masculine or more masculine along this axis. So we can also do this with a female. So here, if you look, neighbor pictures, they look like the same because the modification is tiny. But if you look to extremities, you can see this is very different. So this is the same individual, but we edit his femininity. And now you can understand what was the first animation of my presentation. Here, this is a mandrill which change along this axis of sex. Okay, so to conclude, not really a conclusion, scientific points. If in fields, we notice with correlative study, male mandrills seems to prefer less feminine female. So it was strange, and we want to confirm that with an experiment. So I create an experiment, a looking time experiment, where I show for a mandrill two screens where there is two female faces of the same individual, one more feminine, one less feminine. And it was especially for male mandrills. And with that, I do a binomial model to see what is the preference, where is the preference, and I have significant results which show male mandrills prefer less feminine female. So we can see this on the plot. There is a lot of variables on the model, but I want to focus on this main results. And well, thank you. I can listen if you have any question. If you want to discuss, I also have a poster next Monday. Thank you very much.
Cool. All right, I'm Isaiah Jordan. I'm a PhD student in the Draghi Lab at Virginia Tech. I want to start off by just giving a couple quick acknowledgements. I want to acknowledge my lab mates, my advisor, Dr. Jeremy Draghi, my colleagues, Alex Longcamp and Caitlin Miller. All three of them will be presenting talks later this week, and I highly recommend that you go see them. I also want to give an acknowledgement to Dr. Chris Marks at the University of Idaho. While I was doing my master's, I did some of the empirical work that I'll be presenting a little bit of in this talk. So as far as starting the science, I want to quickly give a little bit of background as far as bacteria and sort of some of their physiology. So it's a commonly known trait in bacteria that in an actively growing population of bacteria, there will be some subpopulation in many species which does not grow despite permissive conditions for growth. And these are really important and were first discovered in the context of antibiotic tolerance because a lot of antibiotics target actively growing cells. Cells which are not growing will be immune to these antibiotics and will eventually be able to wake up and reestablish the population after treatment of antibiotics, which is obviously very bad because this means that pathogens will be able to reestablish their population and cause disease again. This was first discovered in 1942 by Gladys Hobby. It was named persisters by Joseph Bigger in 1944. So the basic idea from this idea of dormancy is that because actively growing individuals that are experiencing fluctuating and changing environments, they will, of course, induce phenotypes that are relevant to the environments that they're experiencing. But once those environments change, they'll no longer have induction of those phenotypes. And because bacteria double in volume before they eventually divide, you will dilute out the phenotype, the relevant proteins by half with each generation. And so because of that, an individual which goes into a dormant state while expressing a given phenotype will not experience this dilution. And if they stay in that dormant state for a sufficiently long time, they can hold on to that phenotype for a very long time. And over time, as the environment changes and the population grows and bulk of the population loses memory of this phenotype, these individuals can establish a much higher degree of phenotypic diversity within the population as a whole. Yeah. So this is all good and well, but is there any actual empirical evidence that these phenotypic seed banks really exist? And I'm going to provide a little bit of evidence that they do in fact exist. So for reasons I won't go into, we were investigating the effects of formaldehyde on E. coli. They were grown in media containing 0.8 millimolar formaldehyde. There's two important things that this does. One is that it acts as a bacteriostat until the concentration of formaldehyde is sufficiently depleted that allows growth. So what that means is that while they're sitting around in this formaldehyde, they're inducing these phenotypes that create formaldehyde tolerance. And then by the time they're ready to grow, all that formaldehyde is gone, and there's no longer induction of this phenotype. And so you can see they wait for about 105 hours before they start growing, which is an extremely long time for E. coli. And that once they begin growing, you can see the gray line, which is overall formaldehyde tolerance in all the population, begins to rapidly decline due to the dilution of that phenotype due to growth. Meanwhile, we have this population of individuals that have sort of a historic tolerance that was generated during the presence of formaldehyde. And because they've been dormant all this time, they don't lose the phenotype as quickly, and they lose it by transitioning back into a growing state and diluting it out, basically. Newly formed persister individuals, however, do not have any tolerance to formaldehyde because they were formed after the formaldehyde has all been depleted, and so they're completely naive to formaldehyde and have no tolerance to it. So based on that, we wanted to kind of create a model to sort of investigate some of the effects of a potential phenotypic seed bank in a sort of different, slightly different situation than the empirical work. So we have a Markov-based simulation with two environmental variables that control the rates of transitions between the different states of the population. We have carbon, which controls growth rates, and toxin, which causes death and also induction. So you can see here, individuals that are exposed to toxin will induce the phenotype, and individuals that grow will lose the phenotype. So there's kind of a tug of war when there's toxin present. And if you are more tolerant, as you might expect, you have a lower probability of death by toxin. Additionally, these individuals can go dormant, and if they do so, then they won't grow and therefore will not lose their tolerance. It's important to remember for our purposes of our model, we are not considering our toxin to be some sort of antibiotic which selectively targets actively growing cells, but instead something that indiscriminately targets cells and only spares cells, or is less likely to kill cells if they're expressing a more tolerant phenotype. So with that, I want to go into some sort of our simulation results. The situation that we kind of created for this was an initial weak pulse of toxin, which causes some death but also induces tolerance, and then a second stronger pulse of toxin, which causes much more death and is likely to kill a completely naive population. We compared two different genotypes in two separate simulations. In red is a genotype which is not capable of forming a dormant subpopulation, and so they will not be able to generate a phenotypic seed bank and will not have any protective effect from that. In black, you can see is the genotype which is capable of producing a dormant subpopulation, and therefore also a seed bank. And so you can see after the first priming pulse, the distribution of phenotypes within the two populations is pretty much identical. However, by the time that the second pulse is about to happen, you can see that there's a big divergence among the levels of the most tolerant individuals in the two populations. So in the population which has no dormant subpopulation, they've completely lost all of the most tolerant individuals, whereas in the population which does have a dormant subpopulation, we still have at least some stash of those individuals that are going to be more likely to survive an intense pulse. And indeed, what we see is that following this second intense pulse, the population without the capability to form a seed bank has gone completely extinct, whereas at least a few individuals are able to survive from the phenotypic seed bank, and therefore they will be able to eventually wake up and reestablish the population. There's two other important results that I kind of want to share with you. One is a set of simulations we did to investigate the longevity of this phenotypic seed bank effect. Once again, you can see in red that the population without any seed bank very quickly loses its memory of the first pulse, and there's no advantage, there's no capacity for it to survive. Whereas the population which does have the ability to form a seed bank can remember the former stressor for a much longer time. And as you might expect, if you increase the rate of spontaneous death that's not related to the presence of any sort of toxin, that reduces the longevity of the seed bank because it's eroding away all of those tolerant individuals. On the right-hand side, you can see the effects of changing the turnover rate. So we've held the rate at which dormancy, they go dormant and resuscitate to be proportional to each other, and vary sort of the turnover rate. And what we can see is that if you have a very low dormancy rate, you don't have very much chance of survival because there's very few individuals in your seed bank. And you can see that in panel B on the right there, that the seed bank kind of also saturates out very quickly with the turnover rate. However, what doesn't saturate out quickly is the effect on residency time. So as you increase that turnover rate, the residency time continuously decreases linearly. And so because of that, as you increase this parameter, the memory of the individuals in the seed bank declines over time. So it's not really advantageous to have a higher rate at which individuals go dormant in this case because they will forget their tolerance much faster. So this leads into the main question for my talk, which is can this effect have contribute to the evolution of dormancy? So we know that, for example, antibiotics present in the environment can definitely select for genotypes which produce dormancy. But what I'm wondering is, can this also select for individuals that produce dormant, genotypes that produce dormant individuals? So to do this, I extended my model to a metapopulation model with multiple patches. The idea is to have sort of the dormancy genotype to invade a population which is incapable of producing dormant individuals. Each individual patch experiences different pulses of a different set of pulses of toxin. They can experience all kinds of low or strong pulses, and this can produce extinction in many of these patches. It does not necessarily, however, produce extinction, and a lot of these will instead induce seed banks or some kind of phenotypic memory in these populations also. And finally, we allow migration between the different patches so that colonies, patches which have gone completely extinct can be reestablished and recolonized. So what do we find for the results of this? Well, from the very preliminary results that we've collected so far, we find that the genotypic ratio of the invader actually declines over time. And this isn't really a terribly surprising result because they have that fitness cost associated with allocating some of their individuals into dormancy. And so because of that, those dormant individuals are losing out on these reproductive opportunities and just basically costing the population a lot of opportunity. So it does make sense that you would have maybe a decline. But also, this is kind of a really complicated environment So could there still be some kind of relative advantage to having this dormancy effect? And what we find actually is no. These dormant individuals do much worse than a genotype, a neutral invader which does not produce dormant individuals. And so what we see is that it's very hard to conceive how this phenotypic seed bank effect could cause evolution of dormancy in a population. So there's one really good reason why this phenotypic seed bank effect might not be having the impact that we expected. And that is because, as you can see in this graph, we're basically way over on the far left-hand side of this graph, where the fraction of the individuals in the seed bank in the entire population of the patch is very low. And so because of that, there's very little chance that any of the relevant individuals will actually make it into the seed bank. And for that reason, it provides sort of no real advantage. If we were to simulate this in higher population sizes, or if the genotypic ratio was closer to fixation, it might have a better chance of having some kind of selective advantage to forming these seed banks. So finally, I just want to kind of present some of my takeaways. The phenotypic seed bank can protect against extinction, at least in like certain scenarios. The degree of the protection is dependent on lots of different demographic and environmental parameters. And finally, so far at least, we don't really have any any evidence for any kind of selective advantage in unpredictable environments. Any questions? Yes. The perturbation, yeah. Possibly. I haven't really had much chance to look at a wide sort of parameter space for sort of the frequency of those pulses or the intensity. If you have more, then yeah, you might increase the chance that you are basically keeping the population, the distribution of phenotypes shifted towards the more tolerant phenotypes, which might increase the chance that individuals that are at the most tolerant state will end up in the seed bank. On the flip side, you might also just, well, for one, right, if you're looking at a very small genotypic ratio and you're hitting it with pulses of stuff that's killing it, that gives a really good chance that you're just going to drive that phenotype to extinction. Yeah. Any other questions?
Still time for a quick one.
Yeah.
So in the case of persistence, I vaguely remember that it's really not dormant or not down with more of a continuum that you have of lifetime and so on. So could such effects help promote the situation of relatively more dormant states? I mean, if you extend the model in that direction?
Yeah, so I mean, you mean where it's more on spectrum where not, yeah, it's not binary. It could be, yeah. There's sort of kind of effect that happens in the model as is, where as they approach the carrying capacity of the environment, they slow down their growth a lot. So that itself can kind of produce sort of a, not quite like we're thinking where there's sort of this distinct state, but where just growing more slowly gives you more memory, basically. So yeah, I could definitely see how if you have a distribution of dormancy or a continuum, right, spectrum, that could also increase the probability of this trait fixing.
Hi, everyone. My name is Wan. I'm a PhD student from the Network Science Institute at Northeastern University. And today I'll be talking about our work on conceptualizing single-cell RNA-seq data as hypergraphs for improved cell clustering performance. We'll begin with the discussion on different network representations or derivatives we can generate from the single-cell RNA-seq data. So in the single-cell RNA-seq data, we have the expression level of different genes measured at the single-cell level, and also called the abundance count data. And the most conventional network representation that we can generate from this kind of data would be the co-expression network. These are the unipartite projections of the abundance count data. And here, unipartite means that we have only one type of nodes in the network. So for example, we can have a cell co-expression network where the nodes are cells, and edges would be the similarity between the cell expression profiles. And similarly, in unipartite projection, we can also have nodes as genes instead of cells, and then in that case, the edges would just be similarity between the gene expression profiles. And then beyond the unipartite network projections, we can also use bipartite networks or hypergraphs to represent the single-cell RNA-seq data. And in this presentation, I may use the term bipartite network and hypergraphs interchangeably because the single-cell RNA-seq data could be represented by either graph, either bipartite or hypergraphs without losing any information. A hypergraph is a generalized graph where each edge could be connected to as many as vertices as you like. So when we conceptualize the single-cell RNA-seq data as a hypergraph, we can let the nodes in the hypergraph represent cells, and the hyperedges represent genes, so that each hyperedge, that is each gene, they could connect to all the cells where the gene is actively expressing. And in this way, we would be able to capture the multi-way relationship of the cell-gene interaction without requiring any transformation. And then next, we'll discuss the limitation of projecting single-cell RNA-seq data into co-expression networks, which are the most conventional type of projection. So if we use a bipartite projection where one type of nodes are the cells and the other type of nodes are the genes, then all the information, like the exact expression level of any gene in any cell, they would all be captured by this graph, so no information would be lost. But if instead we use a co-expression network to represent this kind of data set, then we would only know there are some nodes that are connected because they have a high degree of similarity. But the exact expression level, those information would all be lost in that projection. And secondly, we also know that the single-cell RNA-seq data, they are extremely sparse. Very often we have only about less than 10% of the entries being non-zero, and that sparsity, they could be accurately captured by the bipartite network or the hypergraphs. But if instead we use a co-expression network projection, the graph is going to become fully connected because for any pair of cells, we would be able to get a non-zero similarity score between them, between their cell expression profile. So that means if we use a co-expression network to represent the single-cell RNA-seq data, the network is going to be fully connected. So it's going to be dense. So it's going to be an inefficient representation of the originally very sparse data. And then another problem brought by this projection is the overestimation of co-expression, because we know that the single-cell RNA-seq data, they're very, very sparse, and very often we would merge the cell expression data of different cells. And when we do that, when we create this unified data set, we would include genes that are in every cell's profile. And through that, we would introduce significant sparsity. And this sparsity is going to inflate the correlation between cells because when we are computing the correlation between cells, these shared common zeros, they would just be interpreted as a signal of similarity in their expression profile. But if we know that they are just a result of the induced sparsity, that means that a lot of the signals that we're seeing in the co-expression network, they're actually just a result of the induced sparsity, and these signals, they could be disruptive to our analysis. So the first method we are going to introduce is going to conceptualize the single-cell RNA-seq data as a hypergraphs, because the previously mentioned limitations, they gave us motivation to look beyond the co-expression network because of the previous problems that we just introduced. Here we are going to show the pipeline of our proposed cell clustering approach. So firstly, we are going to conceptualize the single-cell data as a hypergraph where the nodes are cells and the edges are genes, so that each hyperedge in this data set is going to be a gene, and they will connect to all the cells in which they are actively expressing. And then we're going to conduct a random walk process. This is a sampling process that is going to help us to capture the local geometry of the single-cell RNA-seq data. And then through this process, we're going to generate these cell sequences, cell sequence paths. that captures the geometry, and then they will be fed to a neural network that takes sequential input to generate cell embeddings. What is cell embeddings? They're just representations of the cell expression profiles in the low-dimensional space. They would then be used for tasks such as cell clustering and cell type identification. And then the first hypergraph-based clustering method we propose is the dual importance preference hypergraph walk, the deep hypergraph walk. So these methods, we emphasize the importance of both the relative importance of genes to cells, as well as the relative importance of cells to genes. Typically, when we do a hypergraph random walk, we need to consider two probabilities. One is the node to hyperedge transition probability. The other is the hyperedge to node transition probabilities. For example, in this demo here, we have a random walker. This random walker is going to explore the hypergraph. So what it will do is that it's going to select a hyperedge and then select a node in the hyperedge. And then from this node, it will select from one of the hyperedges that is connected to the current node based on a probability distribution we defined. And we want our probability distribution to be able to accurately capture the geometry of the original single-cell RNA-seq data so that we will be able to compute high-quality, accurate cell embeddings. So we have the node-to-edge transition probability and edge-to-node transition probability. And then if you sum over all the possible paths, you'll be able to get the node-to-node transition probability. And we also have a preference exponent incorporated here for accelerated clustering performance. And then we also have a second method, which is going to incorporate the previously mentioned method with the memory mechanism. Through this memory mechanism, we would be able to consider both the hypergraph representation of the data and the two unipartite network representations. So these are projections that we can generate from the original single-cell RNA-seq data. And we will be able to take all these three networks into account when we are computing the cell embeddings. So how do we do that? We do that through a memory incorporation. So when we're considering the edge-to-node transition probability, we would also consider the previous node visited. And we do that by considering the similarity between the previous node and the current node, and similarly for the edges. So normally when we do hypergraph random walk, we consider node to edge and edge to node transition probability. So we only incorporate cell-gene interactions in hypergraphs. But now with this memory incorporation, we also need to consider like the previous entity. So that could be either like the previous node or the previous edge. So through that, we need to also consider like edge-edge interaction and cell-cell interaction. So with this memory incorporation, we would be able to consider all these three networks into account when we are computing cell embeddings. So that way, we'll be able to combine both the local and global level information. So in the hypergraph, we have the local level information because we have the exact expression level of each gene in each cell. And then in these two co-expression networks, we have a summarizing a global level information because what we have there would be the similarity between cell expression profiles or gene expression profiles. And the pipeline is the same as before. From this input, we have cell sequence paths that should be able to accurately capture the local and global geometry of the single-cell RNA-seq data, and then we feed that to a neural network that takes sequential input, and then we'll be able to compute cell embeddings, which are representation of the single-cell RNA-seq data in a low-dimensional space, so that then we can use these for clustering and cell type identification. And then in terms of results, so in figure A, we highlight our results in this purple block here, and all the methods are compared with 5 embedding-based methods, including node-to-vec and PCA, and 4 unipartite-based community detection methods. And we evaluate our performance by average random index and the normalized mutual information. So higher means better, as shown in this bar plot here. Higher value means that the clustering results we predicted, they are more aligned with the ground truth cluster membership. And we can see that our proposed method, the deep hypergraph walk, especially when it is combined with the memory incorporation. It shows the best cluster separability and the highest internal cohesion within clusters. And then moving on, we also evaluate the performance of these clustering methods under different data modularity condition. And in our simulated data, we can control the modularity by adjusting the average number of co-expressed genes per cell type and the number of embedded cell modules. We use this heat map to visualize the modularity of our simulated data. As you can see here, if we increase the average number of co-expressed genes per cell type, then you see a stronger modular structure. But if you, on the other hand, increase the number of modules that are embedded in a single cell RNA-seq data, the modularity is going to decrease. Like here, you can barely see any modular structure. And we also used Barber's bipartite modularity to accurately quantify the modularity of the simulated data we used. So in general, we can see that as the modularity of the data set decreases, or actually increases, then the clustering performance of all the methods are going to decrease because the task is just going to become more challenging. But we show here our proposed method, especially when the memory is incorporated, the co-mem deep hypergraph work highlighted in red, it outperforms all the other methods, especially in scenarios where the data modularity is very, very weak. For example, when the number of embedded modules is like 100, or the average number of co-expressed genes per cell type is like very low, like only 5, we're still performing pretty well. And then we have a more generalized set of results, which is basically the same thing. We compared the performance with 5 well-established embedding methods and 4 community detection-based methods under both strong and weak parameter regimes with varying parameterization. And our methods, they are highlighted in red and yellow. And we still see that the co-mem deep hypergraph work outperformed the other methods almost in all scenarios. And then in conclusion, we proposed the 2 hypergraph-based methods, and one of the methods, they have memory incorporation so that we'll be able to take into account both the hypergraph representation and the 2 co-expression network representation into account when we are generating embeddings. And we also talked about like the sparsity issue that is going to impact the accuracy of the co-expression network that you generated from this data set. And then we compare our proposed method with other methods, and we show that our method performed particularly well in the scenarios where the data modularity is very weak. Thank you. That concludes my talk, and happy to take questions.
Let's do that. Great. Can everyone hear me? Okay. Cool. So my name is Reese Martin. I'm a graduate student in the Tate Lab at Vanderbilt University. I'm going to be talking to you today about some of the work I've done recently studying the relationship between a gene's age and its pleiotropic status. So setting the stage with some background first, I want to establish how we know how old a gene is. If you have not put any thought into this question before, I know that I hadn't before the start of the semester. This may seem like a bit of a weird question, but essentially what you can do is construct a phylogenetic tree with a couple species of interest. In this case, the example I'm pulling from used about 20 species. And then from these species, identify sets of orthologs that are shared between at least 2 of the individuals on your phylogenetic tree. What you would then do is identify how far away you can get from a target species and still find one of your high confidence orthologs. For example here, if you could only find a ortholog in humans and gorilla, then you would know that that is a relatively new gene. It arrived at the latest at the common ancestor between humans and gorillas. But if you could find that ortholog all the way back in Arabidopsis thalianus, so that's the bottom of the graph here, then that would suggest that this is an ancient gene because it's present at the origin of eukaryotes to be shared between both plants and animals. To get at how pleiotropy is measured, we devised what is essentially a gradient of pleiotropy. For those that don't think about pleiotropy all the time, that's that phenomena where a single gene can be associated with multiple traits. The classical examples are that you have 2 traits associated with a gene, but once you get into the data, you actually see that some pleiotropic genes can have dozens or even hundreds of traits associated with them. to answer the questions we posed, we essentially created this gradient where a gene that's associated with a single process, so that would be on the left side, would be lowly pleiotropic compared to a gene that is associated with 2 processes, which would be less pleiotropic than a gene that's associated with 4, for example. We measured processes in 2 ways. We had both biological processes from the gene ontology, so that would be things like enzyme A catalyzes reaction B, and then we also used the string database's protein-protein interaction information, so that would tell you information like protein A is connected to these other 3 proteins with a relatively high confidence. And getting into the data, or getting into how we'd expect these relationships to play out, we would, from theory, think that young genes should be lowly pleiotropic because they've just recently arrived on the scene and haven't had a whole bunch of time to be co-opted by other signaling pathways or traits, and so they're really only involved in 1 or 2 things. Middle-aged genes should be relatively highly pleiotropic, so over evolutionary time, genes are co-opted by other processes, or they themselves co-opt these other processes to optimize organism fitness. So we would expect the pleiotropy and the genome to increase. And then potentially, old genes may see a decrease in pleiotropy as a result of gene duplication and then subfunctionalization events. Or you may see a tapering off of the increase in pleiotropy as these elevating effects are balanced out by the decreasing effects of some subfunctionalization. That's frustrating. These should be violin plots. I don't know why they're not showing up as plots, but essentially these should be unimodal violins following this nice curve. And this data is in humans. Shows that in our youngest age bin, so that's going to be on the left side of the screen, you have on average, less pleiotropy than your middle-aged and older genes, so that's towards the middle of the graph and further to the right. The ticks on the bottom are indicative of the phylogenetic level that each age is associated with, so this would be A is all life, D would be domain, kingdom, then phylum, class, and family. But that's the answer in humans, that we see that this relationship is preserved. What happens when you look at other model species? It turns out that, very frustrating, that these results hold across 6 model multicellular eukaryotes. So in the top right, we have the human data. Then there's mouse, Daniurario, D. melanogaster, C. elegans, and Arabidopsis thaliana data, where if you squint a bit, you can see that this pattern of very low pleiotropic processes on the left-hand side and relatively higher pleiotropic processes on the right-hand side holds. And these data here are from the biological process count. So these are gene ontology data for each of these genes. When you look at the string protein-protein interaction data, a relatively similar story is told, where the young genes have less pleiotropic function than the old genes as measured by the number of proteins that they're interacting with. Although there is no longer this tapering off that was observed. It seems like there's pretty steady increases in pleiotropic function, so that would be protein interaction as you get older. I'm concerned about this next slide. Yeah, this, I'm going to paint a picture with words. To highlight what was supposed to be going on here, the functional groups of genes were supposed to be broken out by their gene ontology processes, with the oldest set of genes being compared to the youngest set of genes in the right and left. And what you were supposed to be seeing, and you'll just have to take my word for, is that metabolic genes are broadly less pleiotropic than cellular processes, which are less pleiotropic than genes associated with development, and all of which are much less pleiotropic than immune genes. So this suggests that there is a kind of functional tolerance for pleiotropy that is segregating between gene functional states. And broadly, matched categories have less pleiotropy than in a younger group than in an older group. So we see the same trends apply within a functional group, and then across functional groups you can get largely different amounts of pleiotropy being tolerated depending on your age group. And then earlier in the talk, I mentioned that we expected that duplication events would reduce the amount of pleiotropy present in the genome. When we went and tested this hypothesis using ensemble data, so we had to drop the Arabidopsis thaliana plots because they didn't have gene duplication data for Arabidopsis. What we found was actually that duplicated genes have more pleiotropic functions associated with them than singleton genes do. This was a bit of a shock to us because, like I said, we had this nice theoretical expectation that you would duplicate and then subfunctionalize, but it seems like at a minimum, you maintain process count and potentially even increase it. And so I want to take some time to discuss why we think that that may be. So the standard expectation going into this project was that a parent gene following a duplication event, and then assuming that the child genes were both maintained in the genome, would very quickly after that duplication, they would both have both functions of the parent gene. Over time, due to loss of function mutations in one of the functions for one of the child genes, you would see functional loss and then kind of an expansion of or an optimization on function 1 between child 1 and function 2 of child 2, which then would proceed to total loss of function in both child genes for the alternate function that they were doing. What we suspect instead is happening now is that you get to this state where you've almost lost all of your function and the function that you're losing out on, and then you've kind of opened up an evolutionary platform to pick up a new function. And so instead of going to just focusing on function 1 and just focusing on function 2, you now have genes that are focused on functions 1 and 3, and then also do a bit of 2, and genes that are focused on functions 2 and 4, and also do a bit of function 1. While we can't exactly say that this is happening in every case, we do have case studies identified where this exact behavior is happening. One of them is the MyoD1 and the MYF5 duplication event that happened in mice, where the ancestral state was capable of robust transcriptional regulation. MyoD1 is still capable of regulating transcription, DNA templated transcription, and then MYF5 has basically no effect on transcriptional regulation, but still shows some small amount of an effect. And if you go look at the gene ontology data for both of these proteins, they both are labeled with that gene ontology process. So one of the things that we may be seeing is just an artifact of how this data is captured, but it still is suggestive of this not total loss of function, and so incomplete some functionalization. With that, I want to thank my lab, and then I want to thank the NIH for giving Dr. Ann Tate money so that she can give me money so that I can work. And then I want to thank the Vanderbilt Evolutionary Studies Initiative for paying to send me here. And I can take any questions.
This might be a cruel question because some of the figures didn't show up, but what happens if you don't log transform? Because your prediction, it didn't look like it had to be log transform.
Yeah, so essentially what happens is that the data gets pretty crazily blown out. So some of these genes are associated with on the order of 1 to 200 gene ontology processes, and the numbers are worse in the protein-protein interaction cases, where you can see some of these pathological cases get to the 700, 800 interactions. And in those cases, these few dots just absolutely ruin the y-axis. So we had to log transform.
Maybe I missed it. Is your data genome-wide, or did you pick genes for each?
Yeah, so these are, I'm going to go back real quick. So we tried to get as many genes as we could. So these numbers up top were the number of genes that we found within each bin. So we were looking at about 20,000, that was the top level, 20,000 protein coding genes. We were limited to protein coding genes because the data set we were using to find our orthologs is only represented with protein coding genes.
Yeah, interesting stuff. When you're looking at biological processes or traits, is there an implicit assumption that these are all independent? I'm just wondering, have you thought about what if you have processes that are overlapping and how you might correct for that?
Right. So for anyone that has ever worked with gene ontology data, you will find that a lot of the times you will get the same trait represented in multiple different ways within a single gene. And so what we did to try to remove that was a bit of manual separation. So if while I was perusing the data, I noticed that you had two very similar or overlapping traits, we would cull that, and then we would apply that culling to all other traits. And otherwise, we only took a single instance of a tip of the gene ontology tree to try to limit that.
Follow-up question. So when you look at the oldest genes, is there maybe is likely that more scientists work on them? So if I have older gene, then I have people in all these different model organisms, and once they know what's in human or what's in C. elegans, then it's much easier. So could it be that your youngest are just not that well studied?
As part of, to answer that question in part, one of the things that we did as a gut check for this data was we plotted the number of NCBI articles where the abstract mentions the gene name versus the number of protein interactions and the number of gene ontology processes that were identified, and that line is flat. There's basically no relationship between the number of processes associated with a gene and the number of articles written about it, which in our mind got to that question of, are we just saying that the overstudied genes are the highly functional ones?
I think it's more likely that in the history of the gene, they acquired new functions over time, or is there just like a bias in that if you're not eutrophic, you're not destined to be around for very long?
I think that it's less that if you're not pleiotropic, you won't be long. I think that it is that over time, the genome evolves in such a way that reusing pieces is just more feasible than novel generation of entirely new function. And so I think that a bit of what we see is represented as just the fact that these genes are around and allowed to be pulled from, increases their pleiotropy, and then there seems to be some kind of ceiling you hit after which you can't increase anymore. And there's a lot of evolutionary theory about how you lose evolutionary flexibility over time with increased functions, so maybe it's something to do with that.
I'm at conflict of interest, but I think I have to call it time.
Totally fair.
Does anyone want to keep me honest? Or I'll just guess. Okay. Okay, so thanks for sticking around. I'm going to be talking a bit about some work I've been doing with computer simulation to investigate sort of a different approach to mutation accumulation and how it would drive extinction of populations. So just as a quick review, I'm talking about mutational meltdown models where we would start with a, not really, okay, start with a new asexual lineage where each individual produces our offspring after some amount of time. Those individuals are accumulating mutations at some rate, and we assume that all of those mutations are deleterious. Everything is bad. So fitness is going to be multiplicative. Everything is like some coefficient that we take away from one, and then we multiply them together by the number of mutations that that individual has. That's going to impact, I should have made W equal to R. I'm sorry about that. That's obvious, but I didn't. I just realized it now. Anyway, the population keeps growing until it reaches some carrying capacity. There's only so much room for so many individuals, so some of those offspring get shaved off. And that is going to keep the population at some size. With the random accumulation of mutations by genetic drift, all of these individual genomes are going to keep picking up deleterious mutations until the reproduction rate gets driven down to the point where the population is going to go extinct. And so this is just like a sort of a cartoon summary of mutational meltdown described from a paper from Lynch. So, of course, there's some limitations to this model. We're assuming that all mutations are operating independently, so they're all having independent effects on fitness with no epistasis. The point has been made that if you do have some epistasis, like synergistic epistasis, that does not have a real substantial impact on the time to extinction, like the mean time to extinction. We're also assuming that the distribution of fitness effects is invariant, that it's not going to change with the accumulation of different mutations. And generally, we assume that beneficial mutations are rare. So you have like the back mutation available for a given deleterious mutation, but we're assuming that there are not that many sites elsewhere in the genome that are going to restore fitness. This is just a quick diagram of empirical measurement of the distribution of fitness effects for vesicular stomatitis virus, and I realize that there's much more recent stuff out there now, much richer data sets, but this is just showing that everything's clustered around 0, having no effect, lots of lethal mutations, and a very small number of beneficial mutations. Okay, but there's also empirical evidence that this is not true, that the distribution of fitness effects is not invariant, that it changes with the individual. So we assume that, I'm looking at something different here, so it's really kind of like difficult to do this, but we assume that most mutations are going to be deleterious when we have a genome that is really well adapted to its environment. So if you're going to change something, it's going to break something that's been working. However, if you bring a population away from that fitness peak, then there should be more sites available in the genome that can accumulate a mutation that are going to build up fitness. And so this is, and I apologize, I'm being self-serving here. It was just easier for me to cite something I'd done like ages ago. This is from a bacteriophage experiment that we did way back in 2005. Where I looked at a library of deleterious mutations, and the ones that like really nailed fitness recruited a lot of sites in the genome that you could get a compensatory mutation at. And so that was altering the distribution of fitness effects based on how severe the preceding mutation was. And again, there's like more recent stuff that I'm sure is much more sophisticated. So this takes us to Fisher's geometrical model, where a population occupies some point in phenotype space. Fitness is optimized at some point in that space, and the probability that the next mutation is deleterious or beneficial is determined by where you're located in that space. So if we are close to the optimum, just as before, most mutations are going to be taking you away from the optimum. They're going to be deleterious. If you're further away from the optimum, then roughly half of the mutations are going to be good. Roughly half of them will still be bad. And so Fisher pointed out way back in 1930 that geometrically the solution was that the probability that the next mutation is deleterious will range between 1 and 1/2, depending on that. Limitations of Fisher's model, it assumes that there is a single fitness peak, a single fixed optimum. There are subsequent papers that look at multiple peaks. So if you have 2 peaks in the landscape, what does that look like? What does it look like if that peak is shifting around with the environmental variation? I'm going to argue that it's difficult to fit this to genetic data because it's a model of phenotype to fitness, and so we have an unspecified map of genotype to fitness, genotype to phenotype. So that makes it difficult to work with, at least from genetic sequence data. There, of course, are other landscape models like the rough Mount Fuji model that also has a fixed fitness optimal genotype, and with fitness decay with increasing distance from the peak. These are 2 visualizations of a rough Mount Fuji outcome that I generated in R. There's like a side-on kind of a wig, and this is a heat map of the same fitness function. Okay, so what I'm going to propose is that we just forget about landscapes for now, okay? The reason is that landscapes are rugged. Like we're pretty sure that they're rugged with respect to genotype. They change with the environment. There's lots of peaks. They're high dimensional. We are just terrible at visualizing high dimensional fitness landscapes. So how about let's forget about it? And we just kind of drift along in a landscape, and we just worry about what is the probability that the next mutation is bad, that it's deleterious. What if we allow that probability to be a function of our current fitness? What does that model look like? And so that's what I'm going to be exploring with computer simulations. The approach I'm taking is I want to be able to fit this to data eventually, so I'm just going to be taking a statistical approach. And this is just like a logistic regression, okay? Because we're talking about a probability, like the probability the next mutation is deleterious. We're going to let that be a function of our current fitness, W, this guy over here, okay? So we've got W, we've got some intercept, we've got some slope coefficient, so this should look fairly familiar to those of you who have used logistic regression. If we set beta to 0, this goes to 0, we just have the alpha, and then probability is invariant. It doesn't change, we're back at our classical model. So, I'm just going to skip over that. Okay, and it's a stats model, so we can fit it to data. Okay, so I wrote a birth death simulation in Python. Why did I write my own simulation? There's other population growth simulations out there and stuff for evolution, because I want the fitness function to vary as a function of fitness, essentially, right? I'm trying to let the distribution of fitness effects change based on what the current fitness is. So we have a whole bunch of individuals. They're all mutating and growing, and the population size is increasing exponentially up to some carrying capacity. And then crashing with some population bottleneck that occurs at some random rate. And if we just let this happen without any special stuff going on for fitness, so if beta is 0, we just have like the classical meltdown scenario. The mean time to extinction declines as we increase our mutation rate when the bottlenecks don't happen that often. When bottlenecks are like really, really frequent, we're really hammering the crap out of the population, and it doesn't really matter what is happening genetically. Okay, so then we add beta, okay, into our logistic model, and so now the probability that the next mutation is deleterious does change with your current fitness. When the fitness is high, the probability the next one is deleterious is also high. And then we let that decay as fitness goes down. So as the birth rate of a given individual goes down, they're more likely to start picking up beneficial mutations under this model. And that has a pretty big impact on the time to extinction. So here we have the classical model, beta 0, and we're increasing beta, and that's, let's say like 4 times, 5 times further out until the mean time to extinction. The thing that jumps out to me, one thing, there's a ton of variation. This is like a highly L-shaped distribution. There are some populations that just keep kicking on for a long time, and then some that don't do so well. The other thing is that there's diminishing returns to beta, so you don't have to have like this weird fitness function. I don't know if it's weird or not, but I might have a hard time persuading some of you guys that this is realistic, but I don't have to, because if we're at about 2, we're still really extending the life of that population. Okay, so last slide. Yeah, this is a silly, simplified model. I'm just trying to learn from what happens when we allow this to happen in organismal evolution. How will we fit this model to data? We would need lots of one-step mutations away from a random sample of genotypes. So deep mutational scanning would give us like thousands of one-step mutations away from a particular genotype, so we would need to do lots of DMS. I have no idea how feasible that is. We'd also have to figure out how to deal with mutations that are actually kind of neutral. Like then there's kind of an arbitrary observational boundary of like what is really beneficial, what's deleterious, and what's like not really doing much of anything. So that's unresolved. Finally, if we wanted to walk a genotype along in genotype space, is this probability autocorrelated? Is it going to be similar to where you were before? I'm going to stop there. I have no idea what I'm doing on time. Well, that's fine. I think I actually kind of rushed things. But, yep, thanks. I'm done. Yeah?
So, is this model clonal, completely clonal, or does it include sex or a combination?
It does not include sex. I have to admit, I work with viruses, so I don't think about that. Yeah. But there's a clonal interference, so I'm not assuming that mutation fates are determined. Like mutations are all like appearing constantly and colliding with each other, but no, there's no sex. Yeah.
Because if you would have sex,
Sure. Yeah. And I don't know what recombination would do in this system, but it's something that I could add it. It'd be kind of a pain too, actually, because there's no structured genome. But yeah, it's a good question. Thanks. Yeah.
So in this model, you incorporate this dependence on how likely you are to have a deleterious mutation based on your fitness. Do I guess, the degree to which it's deleterious also change on fitness? Like, as you get further away, does the chance that you have not only a positive change, but also a large positive change increase?
Oh, like the effect size of the mutation, does that change? No. So something I haven't kind of swept under the rug is that I'm assuming that the effect size distribution is not changing. It's just the sign that, is being influenced.
There's some really interesting work that shows that the farther you away you are from a fitness optimum, the better it is to be taking large steps.
Mm-hmm.
You won't overshoot anything when you're way out there, so you just may be shooting towards the right direction accidentally.
Yeah, cool. I was just trying to start as simple as possible. But yeah, I mean, that's something that could be, I don't know, wouldn't be a logistic regression anymore. Yeah. Yeah, cool. Yep.
You could do this on Fisher's geometric model itself, right? Like if we had explicit principles of functions, then run this random steps. Instead of assuming this logistic model,
Because I wanted to fit this to data eventually, and I'm going to argue that Fisher's model cannot be fit to data in which you're associating genotypes to fitness. Right? You would need to have a genotype to phenotype map and then go from there to Fisher's model. I think Fisher's model kind of gets fit in like we expect this outcome, and so that these are the parameter combinations that are consistent with that outcome, but I'm more interested in just like directly fitting the model to data. But that's why.
so the point related to this is that if you're thinking of genotypes of fitness, so there's the work on global epistasis recently, a couple of years ago, by Desai, where they show that there's a part of epistasis which is actually kind of global and the other part that is history dependent.
Yeah.
Generically. I don't know if you're familiar with this work.
I'm not, and can I follow you up with you later about that? Because I think we're at end of session. But yeah, okay, thanks so much.